We're going to continue with the chapter on DNA replication as well as transcription and translation, and today's focus is going to be on the structure of RNA, its function, and the process of transcription. RNA is ribonucleic acid. Instead of having thymine in its base pair set of A, G, C, and T, T is replaced by uracil. RNA is considered a helper to DNA, and there are three major types of RNA that we're going to discuss in this lecture. If you look at this, the structure of RNA, and think about how we looked at the structure of DNA, there's a lot of differences, but there's also a lot of similarities between the two structures. If you notice, this is single-stranded, not double-stranded, so our bases are not hydrogen bonded with anything normally. We still have the sugar and the phosphate backbone. This is considered one nucleotide. Another difference that you see here is that the sugar for deoxyribonucleic acid is deoxyribose. The sugar for RNA or ribonucleic acid is just ribose, and you see that right here. We see the phosphate backbone, right here. The phosphate backbone is still intact, and then we have our nucleotide bases out here, but instead of thymine, we have uracil in its place. Here's a chart comparison. This is important to know for our end of the year test as well. So the type of sugar that DNA has is deoxyribose, RNA is ribose. The base is adenine, guanine, thymine, and cytosine. RNA has adenine, guanine, uracil, and cytosine. We know that DNA is double-stranded, which allows it to form a helix. Because RNA is single-stranded, it cannot form a helix. There are three classes of DNA, like we talked about a while ago. We have the messenger RNA, and we denote that with a little m, capital R, capital N, capital A. Its job is to help out the DNA get information to the rest of the cell out of the nucleus. So it's produced in the nucleus during a process called transcription. It takes a message from the DNA to the ribosomes. Transfer RNA, or little t, capital R, capital N, capital A, is found in the cytoplasm, but it's produced in the nucleus. It transfers amino acids to ribosomes. And each transfer RNA or tRNA carries only one type of amino acid, so it's very specific. Ribosomal RNA is rRNA. It's also produced in the nucleus and makes up ribosomes along with proteins. So we've talked about ribosomes as being a structure in cells, an organelle in cells. It's mainly made up of ribosomal RNA. Let's look at gene expression. Gene expression requires two separate steps. First of all, you have to have transcription, which takes place inside the nucleus. This is where a portion of the DNA serves as a template for the messenger RNA formation. Then you have to have translation. Translation is going to take place in the cytoplasm, and the sequence of messenger RNA bases, which are complementary to those on the template in the DNA, determine the sequence of the amino acids in a polypeptide. So replication happens when we make new cells. That was the thing that we discussed in the lecture from yesterday. In order for the genetic information inside the DNA to be expressed, it has to be transcribed, taken from the form it's in DNA and put into a different form. Transcription has to occur, and then transcription happens to make messenger RNA. The messenger RNA is going to go out into the cytoplasm, hook up with a ribosomal RNA made up of rRNA, and it's going to be translated from the messenger RNA bases into a protein or polypeptide or a protein with help of the transfer RNAs. So let's look at what happens during the transcription portion. A gene or a segment of the DNA serves as a template for the production of an RNA molecule. All three classes of RNAs are used. The messenger RNA, RNA polymerase binds to a promoter in the DNA. The DNA helix is opened up so the complementary base pairing can occur. The RNA polymerase is going to join new nucleotides in a sequence complementary to the DNA. So it's very, very simple and very, very similar to replication, only let's say this portion is what needs to be transcribed in order to have your body make a particular protein. So this section of DNA on this particular chromosome opens up, and we have DNA polymerase that's going to go down through here and to attach and put the new ones back together. Now these messenger RNAs, individual nucleotides, where do those come from? Those come from in the nucleus itself, so they're floating free, and they come and attach to this portion of the DNA that's being replicated for this particular protein to be formed. It's a little bit more in depth than just looking at making a copy or a mirror image complementary side to the DNA that needs replicated or needs processed for proteins, but it has some other steps in it that are very important. So to process the messenger RNA, the primary messenger RNA contains bases complementary to both the intron and exon segments of the DNA. The introns are intragene segments, but those get removed. Exons are the portion that actually gets expressed. So usually we think exons would exit, so they would be gotten rid of. Exons in this case refer to those that need to be expressed. The introns are the ones that get removed. The intron sequences are removed. A guanine cap gets put on the top of it. A poly A tail is added to the end. And then the mature messenger RNA transcript is ready. So it's been taken from the DNA. We have the copy of the transcribed portion that we need. It has its cap, it has its tail, and it's ready to be transcribed. So here's a simplified version of what goes on once it's transcribed. This is the section that gets transcribed. And once we've had it transcribed, then you have the introns that are going to be removed, and we have the exons that are going to be pushed back together. And you notice that the introns have been taken out. We have the exons, or the ones that are going to be expressed, are in here. We have the cap on the end, the guanine cap, and we have the poly A tail on the end. So this is a mature messenger RNA that's ready to go out through a nuclear pore into the cell, into the cytoplasm, to find a ribosome and to be translated into a protein. This process of replication that we discussed yesterday and the process today of transcription is going to be so much clearer. This is just a really quick lecture to give you basic information. It's going to be made so much clearer when we do the in-class activities that we're going to be doing, the hands-on labs that are going to help us apply this a little better. So make sure that you have your notes written out, that you're familiar with the basics of it like this, and then I'll show you better in class what's going on in those processes in a way that you can better understand it.